postdoc, I'm postdoc fellow from UNICAMP. I'm part of the M3G group. It was coordinated by Professor Leandro Martins. And so I'm going to talk about a project that I started recently. It's about the tryptophan fluorescence. It's an investigation about the tryptophan fluorescence in a DNA polymerase enzyme from classical approach. And basically, the tryptophan is very important for the photochemistry and biophysics as well, because the indole group of the tryptophan is very sensitive to the environment. So it can tell us a lot of things about the enzyme, the environment where the protein is inserted. And so the emission spectra of the tryptophan residues can tell, for example, if the environment is polar or non-polar. Also about the conformation of the side chain, if it's more buried or more exposed to the solvent, and so on. So when the indole group interacts with the light, it is excited to the it goes to excited states, and then it can decay vibrationally, and then decays again to the ground state and emits lights in this process. But it can also decay in non-radiative ways. And these non-radiative pathways are very difficult to be predicted. So it usually requires quantum mechanics methods. And so the quantum mechanics methods are very important to spectroscopy, for sure. They are very good in predict the transition densities and transition dipole moments of the chromophore molecules. And also they met the experiments regarding the maximum of the excitation, taking account the environment, the polarization of the environment and the surrounding environment around the chromophore. But if we use the quantum mechanics, we are usually limited to the amount of frames, amount of structures we can analyze, because they are very expensive computationally. And our idea in this project, in the first stage of the project, is to use methods that you can explore more the classical properties of the protein, use classical methods to infer spectroscopic properties of the chromophore. So one property that you can do this is, for example, the anisotropy. In the anisotropy experiments, they measure the orientation of the chromophore respect to the excitation dipole moments. So the transition dipole moments reorient and then change the orientation after the excitation. And then you can use, for example, a correlation function to obtain the pattern, the same pattern you obtain in the experiments, which indicates the orientation of the transition dipole moments due to the Brownian motion in the solution. So we have a lot of data of anisotropy. The experiments were done with this DNA polymerase enzyme, and so the researchers have choose a residue that is very exposed to the solvent, so it's very sensitive to the temperature. And we have a lot of data to work on and try to reproduce. So the first stage we will do this. We'll try to reproduce the anisotropy decays. This is just a trial results I show you. In the first simulations, in the first analysis, it doesn't match very well the experiments, but I think it's an undersampled set of frames, so you have to simulate more, you obtain more structures from the trajectories to obtain better results. It's clear. Oh, sorry, it's clear undersampled because the anisotropy here goes to 0 within 1 nanosecond, and it's not possible because anisotropy goes to 0 when the protein rotates, because anisotropy takes account the rotational of the side chain, but also the rotation of the molecule, the entire molecule, protein molecule. And within this time scale, it's impossible that the protein rotates. So one thing that you have to keep in mind and pay attention is about, if you are sample, the conformation of the side chain properly. And in one way you can do this is to calculate the solvent the surface exposed to the solvent of the indoor group. And so you can analyze the dynamics of the conformation, and also the SASA itself. It's an important property because if the residue is more deep in the protein structure, it is within a more apolar environment, so it can change the excitation and emission spectra as well. And speaking of spectra, we have a lot of spectra from the same study with this enzyme, and you want to explore this data to construct a kind of a model, because the authors have performed single point mutants with this enzyme, and each of these residues they change, they mutate, are within different protein environments. They are more deep in the structure or more exposed to the solvent. So the spectra are in fact shifted, had shifted or blue shifted with respect to each other, comparing each other. So it's interesting to us. Sorry. So then the second phase of the project, we'll try to construct parametric models using at first classical properties like SASA and electrostatic interactions, given the fact that the indoor group is very affected by the charge, the main charge of the environment. So this, in fact, was done by a former PhD student from your group, but not with this enzyme. You probably you have to parameterize this data. And if you get this model, you can have a formula that you allows, in theory, to obtain a kind of spectra. If you get some many frames from simulations, and then you can infer the excitation, giving a certain value of SASA or electrostatic interactions. You get some a kind of a spectra, emission spectra. So this is the project I'm presenting here. As I said, I started 2 weeks ago, basically, and the project is divided in 3 phases. So the first phase we try to reproduce the anisotropy decay and in water, using the mutants that they studied exhaustively. In the second phase, we use parametric models to try to obtain the excitation, the emission energies of different mutants. In the first phase, you want to include the protein in a more polar or polar environment using co-solvents. Because we want to combine thermodynamics, solution thermodynamics with spectroscopy. So I'm very excited because for me it's very interesting to reproduce data, experimental data using computational approach, and I'm very I hope it will be done well, successfully. Okay, so I would like to thank to the event to give me to the opportunity to expose my project. To FAPES for the fund, UNICAMP for the infrastructure, everyone from the group, and Professor Leandro Martins for the supervision in this project. Thank you.
Thank you. And sorry. It's open for questions.
If you go back that plot of the wavelengths versus SASA, what do you expect to get at? What that line means? That's just a fit, because they're not going to have negative SASA, right?
Negative SASA, no. Yes, but it's I think the curve extrapolated the model. I think it's we have.
So that line is a model, or that's
model.
just a fit on the points?
The line is a model. I extracted.
But how the model goes to negative? That's I'm curious.
Yes, I extracted the data from a PhD thesis from a former, as I said, from a PhD student. Yes, it's a model. It's go beyond. Yes, it doesn't make sense. It does. But yeah, it's a model. It's a model. So we have your interval, confidence interval. So you work in that interval. So.
Any other question? No? Then you thank you again
Okay.
for the presentation.